First speaker is Xuexin Chen, Professor Xuexin Chen from Zhejiang University. And Dr. Tang will give the talk on behalf of him. So let's welcome Dr. Tang. Tampo.
Okay.
You can share the screen with us.
The screen. Hi, Professor Philly. How about my screen and my voice?
Yeah, it's quite good. It's okay.
Okay, I'll start.
We see the presenter mode.
Right. Thank you.
You need to change.
The exchange. Okay.
Yeah.
How about it?
No.
It's okay.
Yes.
Okay. Hello, everyone. My name is Pu Tang from Zhejiang University. Today, on behalf of Professor Xuexin Chen, I will introduce our project about large-scale genome sequencing and evolutionary analysis of parasitic wasps. So parasitic wasps are a larger group with over 110,000 known species worldwide. More or less 20 superfamilies and 60 families of Hymenoptera involve parasitic lifestyle. The mature stage of them parasitic, which result in death of the host. So parasitic wasps are a class of effective and important natural enemies, which play an important role in natural control, ecological regulation, and biological control of pests. The parasitic wasps have evolved into complex and diverse parasitic habits. They can be classified into the variety ways like endoparasitism and ectoparasitism, monoparasitism and polyparasitism, and so on. Such complex and parasitic habits are unique in the animal kingdom, which can provide a good model for us to study the biology evolution of parasitism. The phylogenetic relationship among parasitic wasps at the superfamily and the family level are still unclear. Even two recent papers with big data have not resolved this problem. So we want to use the genome data to understand the evolution of parasitic wasp in two aspects. First of all, by using genome scale data, we can construct a more reliable tree, thus to reconstruct the ancestral states of parasitism. That will help us to answer the origin of parasitism and evolutionary of parasitic ways. And moreover, comparative genomic analysis can also reveal the key genes and new gene-to-gene relationship in parasitoid evolution and genome structure evolution. But until now, the genome of only 25 species in 3 superfamilies and 11 families in parasitic wasps have been published. Our project has planned to sample representative parasitic wasps genome at family level, and that would help overcome several evolutionary problems mentioned above. Now in our lab, we have got 10 parasitic wasp genomes with 3 new data at the family level. 2 of them Two of them have been published in the BMC Genomics. These two wasps are Cotessia vastalis and Didromas claris, which are the parasitoids of the same host, Diadema mouse. But the parasitize different life stage of the host. These two genomes sequenced by the Illumina and the PacBio sequence platforms. Here are some basic information about the two genomes. I'll quickly skip the PPT. Then we constructed a phylogenetic tree based on the 262 single copy genes using ML method. This result consistent with the previously studies, so I also quickly show it. Then we analyzed the gene family expansions and the decreases of the two genomes. The expansion genes of the two families are enriching different pathways. For example, in Cotessia vastalis, the pathway that contain the highest suppression genes in ubiquitin-mediated proteolysis, while in the Didromas claris is alcoholism. In addition, among these expansion families, we found the labs was expanded in all Ephemolidae species. So we choose the labs to further analysis. The labs are metally proteinless in the M13 peptides family, which in vertebrates degrade several peptide hormones and amyloid beta. Meanwhile, our transcriptome analysis shows that 10 lab family members are more abundantly expressed in eggs or larvae. 11 are more abundantly expressed in pupa or adults. Okay, 5 are similarly expressed across stages. Last, we found the two genomes lacked a number of enzymes required for the synthesis. We take the Cotessia vastalis as an example. Cotessia vastalis cannot synthesize 9 essential amino acids, 2 non-essential amino acids. With the help of the host, they can most of the lacked amino acids have been become synthesizable. It indicated the host, the wasp might change the host metabolic pathway to provide the beneficial environment for the development and the survival of their offspring. So based on the genomic data of Cotessia vastalis and small non-coding RNAs libraries, so larvae and the pterocytes, we identify several microRNAs. We first found the 2 of the possible microRNA can arrest a host groups by modulating expression of the host endosome receptor. And this is the first evidence of cross-species regulation by microRNAs in animal parasitism. This study was published in Natural Communication. So this what we did in last years. So we still have some articles in writing. So, but we think we still have a long way to go. if anyone is interested in this project, please join us. Hopefully we can obtain all the genomes we want to achieve the goal of this project. That's all. Thanks. That's all. Thanks.
Okay, so Temple, there's a question in the QA.
Okay, I quit. Okay.
QA. You said do plan to go for species for in the same clade as the parasitic wasps.
I can repeat the question. Do plant gall-forming species fall in the same clade as the parasitic wasps? Is the plant gall-forming derived from insect parasitism? And if you could please keep the answer short. Thank you.
Yeah, I see the questions. Thanks. Actually, several species form for some species fall in the same clade as a parasitic wasps, like some families in the castoidae. But we still don't know the plant gum forming derived from the insect parasitism or not. So we want to sequence more genome data to solve this problem. That's my answer.
Okay, thank you, Dr. Tang. The next speaker is from Professor Fanghao, also from the Chinese Agricultural Academy of Science, and Dr. Nianwan will give a talk for him. And his topic is about the invasive insects progress in China. Welcome, Nianwan.
Okay.
We can see your screen.
Thanks for introduction. Yeah, can you see my screen and hear my voice?
Yes.
Okay. Good. Okay. Good. Okay, I'm Nianwan Yang from Chinese Academy of Agricultural Sciences from Professor Fanghao Wan's group. We mainly focus on invasive species. On behalf of Professor Fanghao Wan, what I'm going to present today is the invasive list of insects reviewed by Genomics. A little bit background information. When talking about biological invasions, the situations in China is that we have more than 600 invasive species, causing a direct economic losses around $30 billion annually. The number of newly recorded invasive species in China in last 10 years is around 60, with 5 to 6 new recorded species per year. And the threats caused by invasive species in China actually reflects the situation globally. When looking at the invasion process, we know it includes 5 phases, from introduction to establish and disperse an outbreak. There's a theory saying that during each step, only 10% of the species could reach next phases, which known as the 10 rule. The core idea here is that only a very small proportion of introduced species could successfully invade it. So the key question in invasion biology is, what factors determine whether a species will be an invader or not, and how to predict a potential invasion? The inherent superiority hypothesis has proposed that invasive species has intrinsic traits superior to those of non-invasive species. And many studies have shown that invasive species do have some common properties. However, because of the lack of genomic data before, the mechanism behind the hypothesis is not well demonstrated. In recent 10 years, an increasing genome of invasive insects has been sequenced with the purpose of providing a genome analysis platform for invasive alien species research, we established a genome and gene database called InvasionDB. The database consists genome data for 131 invasive species, including 54 invasive insect species. This is the homepage of the database, and here is a web address. As a user-friendly database, InvasionDB allows users to search, blast, jailbreak, and download genome-related data. The collection of genome data for invasive alien species, as well as the genetic analysis related to their invasiveness, will help to understand the molecular ecology underlying the successful invasions. So next, based on the database, we try to predict insect invasiveness with whole genome sequencing data and machine learning. By a comparative genomic analysis of a bunch of invasive and non-invasive insects, we firstly identified 24 gene families associated with enhanced defense, allergy, reproduction, chemosensory function, and transcriptional regulation were significantly expanded in invasive insects and likely to contribute to their invasiveness. By using the machine learning algorithm, we trained a classifier to qualitatively predict insect invasiveness, achieving a 93% accuracy. We also established an invasive index formula to quantitatively predicting the invasiveness, and applied both the invasive index and the machine learning classifier to estimate the invasiveness of like other insect species, classifying them as highly, moderately, or minimally invasive. This work provides a new approach to predict the invasiveness of insects at the genome level. Next, I will go into some case study of invasive insects genome. First, the codling moth, Cydia, which is an insect pest of apple, pear, and walnut trees. It originates in Mediterranean Europe and has spread around the world over the last century to reach 70 countries, and it is currently on the quarantine list of more than 20 countries that are under high risk of its invasion. By combining the technology including Illumina, PacBio, BioNano, and high-speed scaffolding, we update a chromosome-level scaffold assembly, as the genome data revealed the unexpected duplication of an olfactory receptor gene, OR3, whose products as we functionally confirmed, enhance the ability of Cydia to exploit pheromones. That's pear ester, which is released by its host plants, and also the 6 pheromones, means it has enhanced chemosensory ability in locating both host plants and mates. We also functionally confirmed that Cydia has rapid adaptation to insecticides, which might contribute to its globally invasions. The results has been published in Nature Communication last year. Another case study for armyworm, which invaded China in last January, is a notorious insect pest for rice and maize. A chromosome-level genome of 4 armyworm was obtained by Professor Kongming Wu and the Yutao Shells group. A genome-wide resequencing of 103 samples and strain identification were conducted and revealed that the population invaded China has a genetic background dominated by American corn strain genotypes. The population invaded China have evolved high resistance to conventional insecticides. However, they're still sensitive to GM maize, the genetically modified maize, expressing Bt toxin. The results have been published in Molecular Ecology Resource in a few days ago. And both of the studies are part of the ILS Genome Program in China, which is a nationwide genome program for 1,000 invasive alien species in agriculture and the nature ecosystem. It is hosted by CAAS and cooperated with more than 40 institutions from China and overseas. The program was launched in 2017, and the first batch of the invasive species for genome sequencing including 13 insect species. Besides codling moths and 4 armyworm, the genome projects of bead armyworm, cotton mealybug, woolly apple aphids, as well as Tuta absoluta are nearly finished. Okay, some take-home message. We have established a genome database for invasive species called InvasionDB. We have conducted comparative genomic analysis and machine learning to predict insect invasiveness at genome level. The IIS 1000 genome program has been launched. The genome of codling mouse and 4 armyworm have been published and explained their globally expansions to some extent. Hopefully, a new technologies of such invasive insects could be innovated in your future. I'd like to thank the IIS Genome Consortium and thank you for your attention.
Thank you for a great talk. Faye, can you hear us?
Yeah, I can hear.
Oh no,
sorry, I need to unmute. Sorry, I didn't unmute my microphone. So there are several things in Nianwan, and there are several questions in the QA talk. I think you can choose one short question for give a brief answer.
Well, maybe we can select one for her.
Yeah, sure. You
okay.
do it.
Maybe, yeah, I see one as my major from, as my understanding, besides the species which are invasive ones as others. Oh, okay. Okay, well, no, the answer, the Mason was asked as her understanding, besides the species which are invasive ones, the others are not, or non-invasive species. That's we have very strict criteria to define the non-invasive species, since we mainly focus on the invasion stage of establishing and expansion population after the introduction. So the non-invasive ones should first have to be introduced to a new region. Second, you should have been studied in the new region and enough detail to know how it impacts the introduced region, and they have been found or documented. They do not have spread beyond the new location or exhibit any other invasive traits in the introduced region. So according to the definition, we only obtained a small number of non-invasive insects species by mining the literatures. Yeah, and by and also solve the unbalanced sample.
Okay.
Is it okay if we go on to questions later?
Because the time's up, and so same things Nianwan, we need to move forward to our next speaker, next talk. So the next talk is from thanks Nianwan. Yeah, can you see? Next talk is from Professor Gongyin Ye from the Zhejiang University, and he will give a talk on the wasp. Let's welcome Gongyin. Hi, Professor Ye.
Yeah.
Can you hear me? Okay. Can you share the window, share the screen with us?
Yeah. 这是最会长的，不要，你到时候。
We can see it. Thank you.
Yeah. Okay.
Okay. Hello, everybody. Good morning for you and good evening for us. I'm Gongyin Ye from Zhejiang University at Hangzhou City near the Shanghai, China. Today I will talk on the genome of parasite wasps and their venom functions. You know, the parasite wasps are the largest group of the order Hemiplerid, with more than about 100,000 identified species and probably maybe 500,000 unknown species, which are content of 10 to 20% of all insects. The standard and significant proportions of its life history attend to within a single host organism. They are also invariably in class or augmented biological control of various insect pests. The parasites are highly devices and species to attack a particular host life stages, such as egg, larval, the pupal, and adult stages. They are highly specialized due to the tight innate relation with their respective hosts and environmental conditions. They can divide into ecto or endo parasites according to their positions. Also, the solitary or geographic parasites according to their egg number, their egg number per hostes. Invasiveness or Canabinitis are also so. This is not for the successful persistent parasites. Wasps have evolved a variety of strategies to overcome the host immune response and adapt to a constantly changing host environment to satisfy the metabolic, national, and ecological needs of immunity parasites. A range of the various factors including venom, poly-DNA various like parasites, and tissues, their impact in the structures. The species and the function of the various factors are different depending on the defined system of parasites, wasps, and its respective hosts. The parasites' various factors, such as venom, have the function to regulate host physiological processes, including the immunity, the development, and the metabolism, so as to protect and ensure the wasp often survival and successful the development in the hosts, and eventually kill the hosts. Some various factors may be applied to improve the biological control of the pest, insect pests, such as by genetically modify of the parasites, their hosts, or some genomical clones. So parasite life status, behavior, and living habits are replicated at genomic levels. Since the first parasites, the genomes were published in 2010 for the Nosonia and the two Kang genesis. And now about from 14 females and 38 parasite waste genomic sequence has been supplemented to NCBIs. So this is provide the valuable resource for molecular evaluations and biological control study of parasites. Given their diversity biologists, parasite waste are now emerged as model system for comparative genomic studies. Now for our study, there are more about the 11 parasite species are reared in our library and are studied. These species are different life traits, such as some is the ectoparasites and some is endoparasites, and some is the solitary and some is grass. Also some with the different hosts. For for them, we are just focused for this, mostly the foremost the pedometed species, just this species. So also some do some the regular species, Cortesia and Habicorn habitus. Now we have the sequence 9, the parasite genomes, of which the size, the vary, from species to species. So our researchers interested mainly focus in the function, the genomics, and particularly for the venomics. For the species, we focus the families, the pedometed species, so as to explore the venom genes for the insect pest control and disclose the mechanism of the interaction between the parasites and the wasps and their hosts. Now to share some of the parasites, the wasp organisms, we have set up wasp the best. This is a genomic resource of parasite wasps, insect host, and the plant. You are welcome to visit our the wasp the best. Today I will introduce our study results results on our chromosome leaf genome imagery of the parasite wasps, Pedomenes purpurina. This is the parasite of the small white butterfly, the Pests left. So the wasp also has shown a high natural control for the smaller white butterflies. So this is the life cycle of the hosts and the life cycle of the parasites. So this is the species genome size is 313.8.1 megabases. So we are so we also have the high C analysis ancestors on the 5 chromosomes, and this is the published in the generally molecular ecological resource. So the genetic relationship between the Pedomenes purpurina and the 12 other respiratory insects used in this study were determined with the genome-wide of 3,399 single copy genes. So we are as the reader flow beaters as the autoglovers. So here is we show the data. This our species is near the Nosonias. So we also have published some special issues in the archives of insect biochemistry and the physiologists. These are about the genomic lift analysis of the crescent methods. These are the 7 papers published here. So now for us, now next I will talk about the venom protein component analysis of these species with the combinations of approaches of genomics, transcrimaticals, and the prometical analysis. So for this species, we have identified the 17 antigens. These are the 17 positive venom proteins. This is divided into the enzymes, the proteins inhibitors, regulations, and the binding proteins, and also some unknown proteins. So for the genomic lipids, we also analyze the venom proteins on the different chromosomes. Now, now we have the genes of more than 14 venom protein or peptides, such as keratin, and the serping, and have been the column and the expressions, and their function have also identified. However, it is pity that some of the venom protein is spread either with the particular genes expression system of ecological expression system. So we cannot don't know study their functions. So we are trying to do it with the RNA the most now to do some. We're also interested in some unknown the protein the functions. So
yeah, 我们只有1分钟了，结束了。
okay. Now here I just search as a case for a serping venom this venom serpents. For this study, we have found this the serping with some different about the 16 isoforms. So we find that their functions. The serping just the functions is the inhibit the host PPO activities. So this protein also is the bind with the host PAP, the inhibit PAP. Then we should inhibit the host manifestations. So thank you for the attention.
Okay, thank you, so Professor Ye, give a very nice talk. And there are several questions in the Q&A panels, but because of time limitation, and maybe Professor Ye can give the answer in the Q&A or slides.
I apologize in advance if I do not pronounce the names right. So
yeah,
first we have Professor Cheng Sun, who will talk to us about bumblebee genomes. So if you could please share your screen. Thank you. Yes, it works.
Hello, everyone.
Yes.
Yeah, this is Cheng Sun. I'm from Institute of Apiculture Research, Chinese Academy of Agricultural Sciences. I'm very happy to be here to introduce our pretty recent work on genus-wide comparative genomics in bumblebees. So at first, let's see some more background information. So something about bumblebees. So bumblebee is a group of pollinating insects in genus Bombus. And there are about 250 species worldwide, which could be placed into 15 subgenera. And then let's see their importance. Actually, bumblebees are important in many ways. Today, I'm gonna see their importance in agriculture. So bumblebees are excellent pollinators of greenhouse vegetables, especially for tomatoes. And the use of bumblebee could decrease the cost of labor and improve the yield and the quality of the fruits. So at present, there's over 1 million commercial colonies were sold each year worldwide. So the next question, why we sequence many of them? Actually, and definitely, it's not just for fun. So China has the largest greenhouse area around the world, which is almost 2 million hectares. And therefore, there's a huge demand for the pollination of greenhouse vegetables by bumblebees. The good news is that China has the most bumblebee species. From this figure, you will see the distribution of bumblebee species around the world. And you will see China is the global hotspot of species richness, with half of the world's bumblebee species, and they representing 14 out of the 15 subgenera. But which is awkward that so bumblebees used for pollination in China are mainly imported. So however, as it was found out in many countries, so species importation could lead to biological invasions. So it was strongly suggested that only local bumblebee species should be grown for commercial development and used within their native range. So we have to rear native bumblebee species to provide pollination services for greenhouse vegetables in China to avoid biological invasion issue. So to use the up-to-date genomics based methods, we definitely need genomes. However, although there's over 120 species in China, no reference bumblebee genomes is available yet. So we need to generate reference genomes for representative Chinese bumblebees. So which species need to be sequenced? So to broadly sample genomic diversity, we choose species based on the following criteria. For example, there are phylogenetic position. We want to include species to span all the 15 subgenera. And also we will consider there are species trees and their geographic distribution so that we could have species that having contrasting habitats. And also we will consider their potential to be employed for pollination services. So at the last, a total of 17 species were selected for genome sequencing. And they span all of the 15 subgenera and including the social parasites. And this is a collection size. This collection size actually includes almost the most beautiful part of the work. And four bumblebee species were collected at extremely high evolutions, especially at one collection site. And our collection team must wear oxygen mask to capture them because the evolution is more than 5,000 meters above sea level. And also there's 3 species that only distributed in China. Let's see the results. We use a sequencing and assembly method that's pretty suitable for insects. And finally, we generated 12 scaffold level and 5 chromosome level genome assemblies. Those assemblies have pretty decent continuity. And we use a Basco analysis to evaluate those genome assemblies. And we found there's a remarkable high completeness of those genome assemblies. We use a maker pipeline to annotate protein coding genes in those genomes. And we achieved a pretty high quality of annotation for protein coding genes. So right now we have generated a genus-wide high quality genomic resources for bumblebees. So we could do some genus-wide comparative genomics analysis. So
right. 3 minutes left. Sorry, 3 minutes left.
Oh, okay. So from the genome scale phenology analysis, we found a very high level of discordance between gene tree and species tree. And further analysis indicates that incomplete lineage sorting should drive the observed gene tree discordance. So we will suggest in the future gene-based molecular evolution analysis in bumblebee should use gene level phylogenies. And our chromosome evolution analysis indicates that social parasites have a much higher chromosome number than those of the others. And this high level chromosome number results from a major chromosome arrangement. And intriguingly, we found that the rate of chromosome evolution in bumblebee is much lower than those of fruit flies and mosquitoes, although bumblebee have a high recombination rate. And there's a genome size variation across bumblebees, and it was transposable elements that drive genome size variation. We found evidence that those Ps have been domesticated in bumblebee genomes to form novel proteins and regulatory sequences. And the gene content turnover analysis indicates the rate of gene gain and loss in bumblebee is pretty similar to mosquito and fruit fly. And gene structure evolution analysis indicates that there are small gain than losses of introns in bumblebee. This is unlike fruit flies and mosquitoes, where intron losses dominate. the intron-extron boundaries within bumblebee genes are pretty stable. And we found the boundaries of bumblebee genes could be flexible with over 300 genes undergo stop codon read-throughs. And our results support the prediction that insect species have abundant stop codon read-throughs. And our codon usage analysis indicates there's no evidence for selection on optimal codons in bumblebee. This is in contrast to fruit fly, but similar to mosquitoes. Codon usage bias in bumblebee was mainly driven by AT content. We also have identified non-genes involved in high elevation adaptation in bumblebees, and they involved in food searching and metabolism. And a brief conclusion, we generate a genus-wide high-quality genome resources for bumblebees. This set has been deposited in Big Data Center and is uploading to NCBI and will be released after publication. And we reviewed how bumblebee gene and genome have evolved across the bumble's phylogeny. Those genome resources will facilitate the breeding of native bumblebee species in China, and also I think they will contribute to the bumblebee community worldwide. Thanks all my collaborators, and thanks for your attention.
Well, a big note of thanks to Professor Cheng Sun for that excellent talk. I think there are already questions popping up, and we will go over them at the end.
Oh, right.
Yes, we can see it. Thank you.
Okay. Thank you all. And my name is Weihua Ma, and I'm a representative of the Rice Insect Pest Genome Consortium. The consortium was built by 3 groups from 2 universities, and one of the is academic institute. So totally are 6 professors of them. And the goal of our consortium is we want to explore all those key rice insect pest genomes in China, especially in China. And we want to try to identify their key factor in the rest and host, the rest and the insect co-evolution, and we want to find the really key factor that would influence their evolutionary tendency, and then we can use this tendency to make a very specific control strategy for really great agricultural condition of the environmental risk or something like that. So this is the goal. And right now the major rice pests in China are including 3 permanent hoppers and 2 Leptarian. And they all infect here in the plant hoppers are sucking insects. They infect from outside, and the two borrowers they infect in the middle of the stem of rice, so they will give a big loss of yield in the rice production. What we focus on is to use those genomes, and we want to analyze the most important factors in the overwinter ecology, or how they can make those kind of the lifestyle of they stay in the stem, and why they sucking outside, and why they go to the migrate, anything about that. And we really want to find which one is the most important factor in the genome and affect their ecology and evolutionary character. And we use this molecule, into adaptation of the agriculture pest control. And among all of the topics, I will use the animal antifreeze as an example, because insects are ectosomorphic animal. They don't have clothes, they don't have air conditioner when the winter is coming, but they can survive the winter in most of the planet. And the hypothesis for animal antifreeze mechanism is when the cell or intracell they have ice there would be a lot of chemicals or proteins were produced to protect the cell around them from being dead. This is a picture, is a pioneer work of a wood frog. They put the frog in -7 centigrade, and as the time going on, the ice increase. So I means ice increase, and dark is ice. And this is the first picture. We have the evidence that there really is ice inside an organism. And take the Callosupersalis as example. The Callosupersalis, the most they can survive in the 45 degree of latitude, northern latitude, and there's also some data showed that they can live in -10 centigrade, and also if you put it in 0 degree first, then they can live in for a very higher level. So it's a good. This organism is a good ideal material to analyze the antifreeze mechanism. We collect those samples for transcriptome sequencing, and we set a code combination and a non-code combination, and diapause and non-diapause, because some people said diapause is a really important factor to induce the code tolerance. So we pick up those samples for transcriptomic sequencing, and we analyze those genes upregulate. This is a glycerol. It's a very famous cryo protectant, and was functioned in many animals. They said the cell will build a glycerol to protect themselves from freeze. So we check the classical pathway for glycerol synthesis, and we found several genes. One is here, one is here. This one is a very important pathway to make three carbon chemical, and one is here, and they are showed good showed the detected overexpression in the code situation. This is the evidence that the Callosupersalis, they can use glycerol as well as a cryo protectant. And then where those carbon results from, we also find some of those enzymes like amylases and glucanase and maltase. They can make glucogen for the carbon resource, function carbon resource. And then it's very interesting. We found another gene. It's a triacylglycerol glycerol lipase. We found 4 of them are very overexpressed when the code is coming, and they can build the glycerol from TAG directly. They don't need to use the glycogen anymore. So in this insect, they use 2 pathways to make a glycerol. And therefore, they also have a mechanism of the cell membrane protecting. One is a fatty acid, and because we found this fatty acid synthesis is very highly induced when code is coming. So they have we believe they have those this pathway for cell antifreeze, and also they have another little molecule, the trichalose, we can use to protect the cell membrane. Besides all of those membranes, we also have find some other molecules like HSP, and this is the terriferin catalysis as the antioxidases, and the water channel and the storage protein are also functioning in the process of antifreeze. This will take one of the TAG lipase gene as an example. We knock down the TAG lipase.
One more minute.
Okay, thank you very much. And then we find a higher mortality when the time is going up. So that means once we find a key factor of the overwintering strategy of this insect, we may use this factor to develop new pest control strategy. And for a conclusion, we will be short. The antifreeze mechanism of Callosupersalis will be when early freezing, there will be ice outside intracellular, and when freeze going on, they will conform a lot of different chemicals or proteins and fatty acids, and they to protect the cell to be from that. And in the next plan, we will sequencing more rest pests like CM, they infect on the leaf of the rice, and also SE, the Invasiveness, and also there are two stem borers in the future. Those are, I will say, thanks to the people to helping to assemble those genomes and do the analysis of transcriptomes and the biological characteristics. Thank you all. Thanks.
Thank you, Professor Ma, for keeping on time. And to stay on time, let's just move on to the next speaker. You can see your screen.
Yes. Can you hear me?
Yes, we can. Yes, we can see the presentation mode. Thank you.
Okay. Thank you, ladies and gentlemen. Good evening in Beijing. So first of all, I would like to thank the invitation from the conveners of the symposia. And before my talk, I think my Chinese colleagues gave us the very, very important and detailed information about the insect genome in China. So I would like to take the opportunity, give you a very brief introduction about the insect genomics in China. So my talk includes 4 parts. First one, I would like to share some data to indicate the status of the insect genomics research of China in the world. So since 2000, Chinese entomology research experienced tremendous development. Chinese entomologists contribute the 10% entomology papers in the world during 2006 to 2015. Based on the Web of Science, the number of the publications of the entomology in China has increased 10-fold in the past 10 years. The number of the papers and the citation in international peer-review journals from the Chinese entomologists have ranked number 2, and the number of the most cited papers from China ranks number 3 in the world. So look at this picture. So you can find the research paper numbers in insect genomics as one of the entomological disciplines also have the rapidly increased, especially in the last 5 years. About 20 insect species per year are sequenced worldwide. So the Chinese entomologists totally contribute about 27% papers in insect genome in the world. So this picture shows you the numbers of the published insect genome papers from China. So you can find at least 60 insect genome projects have been completed by Chinese entomologists. And the numbers of the new genome project is dramatically increased in China in the last 4 years. In 2019 and 2020, Chinese entomologists published about 20 and 12 and 15 insect genomics papers, and at the same time release the 12 and 40 database of insect genomics into NCBI and other public database respectively. So until today, I think we have the 66 insect genome data released. The half of the data deposit in NCBI, another half deposit in the other public database, such as the Giga database and other database. So at the same time, the 70 pollinators genomes published in bioarchive and released in the National Genomics Data Center database. In second part, I would like to show you some data about the highlights of the insect genomics in China. So I just selected some research paper which published the international peer-reviewed top journals, such as the Science, Genome Biology, Nature Genetics, Nature Communication, Nature Ecology, and Evolution, and so on. So I'm sorry I cannot mention all of the excellent job here because the time scale is very limited. So I think the Chinese entomologists completed the first insect genome, except for fruit fly in 2004. On the other hand, we completed the biggest genome of the insects in size is sequenced so far, such as the locust genome, which has the 6.5 gigabase. At the same time, we will challenge another biggest genome of desert locust. So we completed the first whole genome sequence of the worldwide Lepidoptera insects, diamond bark moths. So this work includes the whole genome sequence and the resequence of the different population in the world. Okay. In the third part, I would like to show you the main research teams of the insect genomics in China. I'm sorry I cannot show you all of the group in China, but I just introduced some major teams. In this part, I would like to show you the 3 teams from the Chinese Academy of Sciences, 2 teams from the Chinese Academy of Agricultural Science, and I would like to show you another 8 teams from the universities. The first one is my group. My group focuses on the ecological genomics of the locust. So we want to answer the ecological question by using the genome tools. Another group is Huang's group in Shanghai, which decodes the genome of the black soldier fly. Zhang's group in Shanghai, which focus on the genome evolution and environmental adaptation in insects. 还是不能动，不能切换。Okay, in Chinese Academy of Agricultural Sciences, I would like to introduce the Wines group in which focus on the invasive insects genomics. Another group is Zhang's group. They focus on the genomics of the vegetable insect pests. And more groups come from the Chinese universities. First one is Xia's group. He also worked on the functional genome project of the silkworms. The Ye's group in Zhejiang University, just before he showed his excellent work. And Li's group in Zhejiang universities his group focus on the rice insects and invasive insect pests. And Chen's team in Zhejiang universities, this group focused on the parasitoid wasps genomes. This group in South China normal universities. The Professor Li is a very energetic expert who work on the several species of the insects, such as the silkworm, cockroach, and Drosophila. Professor Wang's team, in fact, he worked on the phylogenomics in other animals. At the same time, he also worked on the butterfly phylogenomics. The Zhou's group in China agricultural universities, he worked on the insect phylogenomics. And the youth group in Fujian agricultural university, as you know, he published two papers about the diamond back moths. And finally, group is the Chen's group. He worked on the genome research of the mosquitoes. So here I would like to summarize my talk. I think the Chinese entomologist makes an excellent contribution in insect genomics in the world. At the same time, they continue to work on the more species of insect genomics, such as this table show you the ongoing project of the insect projects. In addition to the genome study, I think the Chinese entomologists will extend the more wider genomics study, such as the genome evolution and the phylogeny of the relative species, gene function and mutant line establishment by CRISPR-Cas9 and other gene editing method. Epigenetics, including the long non-coding RNA, DNA methylation, and histone modification. And some scientists work on the metagenomics of the microbes in insect guts. Multiple trophical interaction at genomic levels and phenotypical plasticity regulated by the genes and genomics. I think I should stop here, and I would like to express the thanks for Professor Li Fei. He prepared some of the background material for my talks. That's all. Thank you for your attention.
Thank you, Professor Kang. This was an excellent presentation which helps the attendees from all over the world get a good overview of all the active research projects in China. And let me just start off with a question for you, Professor Kang. This session was where we are trying to develop connections. What other initiatives would you like to see, or have you planned to encourage further interactions and eventual collaborations between arthropod researchers in China and rest of the world?
In fact, the Chinese entomologists have so many collaboration with the foreign experts. So some foreign experts, in fact, take part in the insect genome project in China. So you can find some of the papers in authorship. So you can find so many international experts take part in the our project. In the future, we are welcome to the more foreign experts to join us, so we can extend our research experience. At the same time, we want to sequence more species of insects. So our major purpose for insect genome research, first one, we want to reveal the scientific phenomena and story. Another one, we want to explore some more important information to develop the tool for the pest management.
Thank you. And two related questions. One is, how are species selected and prioritized for funding so that they can be sequenced? Is there a mechanism?
Because recently, with the economical development of China, so we can got some money to support our work with the insect genomics project. So in China, we have the three channels for insect genome project. First one is the Ministry of Science and Technology. So in this channel, the major support emphasizes the basic and applied studies, which linked with the national needs. Second one is NSFC, which support the very pure scientific and basic study. So we can get some money to study the insect genomics project. The third channel is the Chinese Academy of Sciences, so which can provide a stable financial support. So we can use the channels to support the insect genome study, but we can select the different insect species depend on the different channels demands.
That's excellent. And this, the last question, and then I will hand it over to Fei if he has any more, is to understand how the data sharing is the vision is, because do you feel that, or do you think the China National Gene Bank database will be joining the INSDC, which is the International Nucleotide Sequence Database Collaboration, where we have GenBank and CBI? Do you think that that will be a connection?
Yes, it is an excellent question. So in fact, Chinese entomologists observe the international rule of the data sharing of the insect genomics research. So usually the Chinese entomology cannot release the data until the paper accepted and published. So I think Chinese entomologists observe the common rule, which is similar to the other countries' experts. At the same time, in China, we have the several database resources. So recently, Chinese entomologists usually deposit the data in the different database, including NCBI. So this is why some foreign experts cannot find the data because in the world, there are several databases, including NCBI. So I think the foreign expert should contact the Chinese entomologist, ask them where the data which are deposited. So I think we would like to share our data. But in some entomologists are used to be released the data after the paper published.
So some of this might...
in China, we have the database, the BGI database, and the national genome database. So this database is also open and a public database.
That's a great point. So do you think these databases, which you just mentioned, the BGI one and the National Gene Bank database in China, will they be joining the consortium of INSDC databases where we have DPBL in Japan and NCBI?
Yeah.
Great, thank you. Feli, did you have any? Thank you. Feli, did you have any more questions? And otherwise, we can.
Yeah, thanks, Professor Kang. Give a very nice talk, and I have no more questions.
Okay, great. Yes, let's go through questions for the other speakers. We are at almost the end of the session, but we'll continue with the discussions for at least 10-15 more minutes. So please hang around. We will get to your questions. So let's start from the top. So this is for Dr. Purang. Do you see a general pattern of gene loss or gain depending on the type of the host of the parasitoid wasp?
Thanks for the questions. Generally, we found many conserved genes tend to lose during parasitism evolution. Actually, one of the lab members have submitted an article on this and this article is under review. So hopefully this paper, I think, will come out soon. If they're interesting, this where you can download the paper when they publish it, I think.
Great. We do have a channel for preprints and papers on Slack. So once the link is available, please post it so that we can all access it.
Okay.
Thank you. All right, I have a question for Dr. Wang Ji, but he is not online right now, I think. So we'll move on. A question for Nianwan from George Benoit. Does the invasive predication take into account agricultural practices or stress tolerance related to survival in specific environments for your invasive species?
Pardon?
Okay. So when you call a species an invasive species, does that take into account agricultural practices or stress tolerance related to survival in those specific environments, for example, for that crop? And Josh, please jump in and rephrase if I'm not doing it justice.
I can rephrase. It was probably not written that well. Mainly so you use the genomic predictions to look for if these are invasive or not. Do you take into account like environmental conditions in specific areas, or what sort of plants are grown in those areas that might be that the specific invasive animal may actually become a pest on?
Okay, if you only if the insect pests, the environmental have influenced the invasive species and has already make the genetic changes in the gene family level, then it could be predicted by this approach. There's other models, ecological models, to predict the invasive list, including the environmental and also the other factors. But if it is not influenced the invasive species in the genetic level, it cannot be predicted by this approach. However, most of the invasive insects, they have very rapid adaptation to the environment. For example, if it escapes from the lateral enemy, and if it changes the allergy which is in its original locations to defense, to for example, to reproduction or to nutrition requisition in the newly location. So that could be predicted by this approach. Yeah, that's my answer.
All right, thank you.
We have another question for you regarding the duplication of olfactory receptor genes. Is it because of pest or ecological reasons? What do you think is the evolutionary driving force? And this is a question for Nianwan.
Okay.
Sorry.
Sorry. Once again, sorry.
Sure. Regarding the duplication of olfactory receptor genes, do you think it's because of the host pest or ecological reasons? And do you think there are any evolutionary driving factors for the duplication in olfactory receptor genes?
Yeah, I think that's evolution driving that's has the duplication, because that's the different set of OR3. They have different functional tools to respond to the host plants and host pheromones of the host plants, and also the 6 pheromones. That's different set of functions. I think that's the evolutionary stress for that.
Great.
That's it.
All right, thank you. So now we have a question for Dr. Cheng Sun. If you could unmute yourself. This is for Cheng Sun. Can you hear us?
Yeah, I can hear you.
Okay, great. So this is a question from Marianne Goldsmith. How do the general observations and conclusions so far about bumblebee genome structure and evolution compare to Apis mellifera and other Apis genomes, in addition to fruit fly and mosquito?
Oh, so actually, I saw this question and I replied to her, I think. Yeah,
Okay.
yeah, we didn't prepare it. Yeah, thank you.
Okay, great. So, well, let's find another question for you. So it's great that you mentioned the methods you use for sequencing and assembly to produce these chromosomal level assemblies. Are you planning on upgrading the older genomes at some point? And if so, with what technology would you do it now? And this is for Cheng Sun again.
Oh, okay. So I think the most difficulty we meet is that we cannot collect enough samples because they are mostly wild bees. It's very hard. So if we can collect enough samples, I think we will use Hi-C to make chromosome level assemblies for all the species, so we can understand how the genome structure changed.
Great, thank you.
Yeah.
This is a question for Weihua Ma. If you could please unmute yourself for Professor Weihua Ma.
Oh, yes.
Yes. What kind of biological questions will you focus on for better understanding of the interactions between rice and its insect pests? For example, defense genes or developmental genes?
All right. Actually, I'm not very clear in mind what kind of ortholog I will find, but but I know what I want to find is something about the host adaptation or the key factors between the defense and anti-defense between the rice and its pests. And we really wish to know why they can co-evolve such a long time and they stay together, especially for some kind of the monophagous insect, and like brown hopper, they just eat rice. Why they do this? And when do they begin to do this? I mean, maybe some million years before. And how can we find this? And if we really can find this such a great factor, we can use it to control the pest. And we can, but I'm not sure if it's good because they eat rice only, so maybe not good. So, but for a human being, this is good, right? Okay, thank you.
Thanks. Yeah, that's a nice example of having access to the data in a central database where all these resources can be mined together, where you can look at host range and specificity. So yeah, we all hope that the databases that were mentioned earlier, they would at some point have a crosstalk between NCBI, Gene Bank, and EMBL, so that all these resources are in a central place. All right, next.
Yes, I do have a question because compared to those computational guy like you, I'm a biological guy. So we really wish to know in the present status, and I mean in the techniques which was improved like this today, can we really to search for the popular, the popular orthologus in any kind of a small scale of organisms? I'm not sure if I express my information. I mean, like I asked a question for, like we said, we are sequencing rice pests. Maybe because I focus on rice. This is a host, but some other people they say they are sequencing the vegetable pest, and some of the pests maybe they are insects. Okay, they are insects. Yes, they just the host spectrum. Can they have we use those kind of classification of a host, and can we find some popular orthologus which can reflect their host spectrum, or they're like anti-fries and their dipoles, something like that? Can we do this right now from a bioinformatics view?
So I think OrthoDB, where I think Rob can add, is probably one of the most comprehensive resources for orthologs, which keeps on getting updated regularly. But yes, Rob, did you want to add something?
Yeah, so it's difficult to keep up with all the new species that are coming along, but you know, at the base fit is the all against all sequence comparison. And currently, OrthoDB has about 170 different arthropod species included in the current release, which is possibly the largest in terms of any arthropod representation out there currently. And that's the first sort of port of call to go to when you need to find orthology data to begin to start to interpret all the genes in your new genome.
So we still focus on the annotation. I mean, if we still depend on the annotations or for this. Okay, so maybe we find some like domains like in the genome in specific domains. Like they maybe include a lot of genes, but they have some kind of popular character. Just my thinking. Because I'm not a computational guy, so. Just a joke, okay?
Yeah,
yeah.
I mean, I think this is a general theme, you know, not just in this session, but across all the sessions, whereby each new genome that we sequence and assemble and annotate adds to the bigger picture of what we can get out of it. So as we improve the species sampling in each and every single arthropod clade out there, we are able to get more out of the genomic data. So that's why it's so important to think about things like standards, think about things like sharing, think about things like common best practices, which is exactly why, you know, these kinds of questions and topics have been raised time and time and again at Arthropod Genomics symposia over the years, but in many of our discussions, I can see Sue is nodding a lot there. Yes.
Yeah.
Okay, thank you. Thank you so much.
Yeah, essentially, I mean, it depends on what you're sequencing, but even here, let's say, when we sequence a new genome, essentially, we have to go out to all the databases individually, collect relevant related genomes, and do the orthology analysis de novo, essentially, to figure out what is the most relevant comparative set for our newly sequenced genome. And I think Rob and I are circling around the same issue of data sharing and standardization, where if all the databases start talking to each other, then this data collection and analysis will help everybody. Sue, did you have anything to add?
I was just going to say that at every single Arthropod Genomics Symposium, we've always had at least one session or workshop on what are the best practices for doing a genome, starting from sample collection to sequencing technology to assembly algorithms to annotation practices. And I think the biggest one at the end of that is always the data collection and the data archiving, the data sharing, and the data collection for each new group.
Yeah, and that's an interesting. Sorry, go on.
Yeah, I think it's also a big problem question for the Chinese groups for data archives, data sharing, and also especially for data sharing. It's a good. I think it's better maybe not only for Chinese research, and I think all over the research groups all over the world, and we need to have a discussion about the database sharing and data sharing. Some interest sharing group, and maybe just at the beginning of the genome sequencing, maybe some groups maybe will work together and how they share the data. And then at the mid stage, and then this data will be released to a broader, more people and more person. And then at the end, and then this data will be shared to the public available to all researchers. And I think at the different stages, and how this data can be shared by step, and just to protect the person, protect the right of the person who sequences this genome, but also can push and be helpful to the society of the science. I think we need to work out our rules for all over the world for us. I think it's better for us.
I think that's a great point. And Monica Paulshaw, who runs I5K, we were discussing an idea of doing a genome project management workshop, essentially like what you were saying.
Yeah, good idea.
Right? If some lab has the DNA, they have access to funds for sequencing. How do you most efficiently plan for sequencing, assembling, annotating, releasing the data, all the 4, 5, 6 steps in that pipeline? And these will differ for each organization based upon their funding, access to facilities, and of course personnel, because as Dr. Ma was just saying that some people may not have the mathematics support that they need, but there are lots of other people who are happy to collaborate, and that's usually what happens at Arthropod Genomics Symposium sessions, where these people get together. So we will reach out to everybody, and we will try to set up at least a Zoom meeting to start with for this genome project management workshop not during this meeting, but at least we'll reach out to you, and then we'll try to set something up soon. If you have specific ideas or needs, please communicate those to the organizing committee, and we will try to address everything as much as possible.
Thank you.
Great. So we have gone over by a bit, but I think this discussion was very important, and that was the primary goal of having a special session dedicated to China. And maybe next time we can have more researchers from Asia participate. And if we can be a little bit more friendly, it's in terms of time. I know it's very late for most of you. Is there anything else we should cover, Faye or Rob?
No, I think it's been very, very good. So thank you everyone for keeping on time as best we could, and this great discussion at the end. Of course, we asked the speakers, not now, it's okay, it's late, we know, but at some point tomorrow to please go onto the Slack to start to reply to some of the questions that we didn't get round to discussing today.
Yes, we will be posting these questions to the chat speaker sessions channel. So please go there, and we will try to address it to you as much as possible so that you know which questions are for you.
Thank you to everyone.
Thank you. Good night.